And I bet you can see how much.
So just want to welcome everybody. Thank you all for being here today. I'm Michelle Janelsons, and I am joined by my colleagues. Adam, you got to say your last name because I'm going to. Sespakowski. And Colleen Netherby Winslow. And we're going to be talking about this really innovative shared resource that we have here at U of R and Wilmot Cancer Institute. So this is the Human Biophysiology Shared Resource. Right. Oh. And why is it not letting me advance?
Because you're on Zoom.
There we go. Okay. So just as a reminder, this is the second Tuesday of the month. There's going to be 2 more in the series for this year. One on November 12th, which is also an interesting topic on reducing barriers for data analytics. And then one on December 10th, which is Shared Resources 101 for community members. So please, we encourage you to also attend those as part of this Lunch and Learn series. And also, if you know others that might be interested in these topics, then please help spread the word. Also want to thank sponsorship for this series, very generous support. So this shared resource has 3 main aims. One thing that we do is we consult on design of interventions, assessments, and execution for cancer prevention, control, and supportive care trials. So really across the continuum. A lot of our work is really focused on the supportive care and cancer control end of things. However, we're also involved in some prevention aim studies. We do multiple assessments. You're going to hear more about them today from the human performance side of things that Adam's going to talk about, and then Colleen's going to talk about some of the human biophysiology aspects. And then we also work together to integrate that data. So just some examples of some of the types of studies that we have helped collaborate with is in both local trials in our catchment area and also nationwide trials all throughout the US in both cancer prevention and control. And we have a longstanding legacy of supportive care work in symptom science here. And we also have a longstanding legacy of this integrated physiology and biomarker assessments by combining the expertise of our 2 labs. So the CCPL lab, which I direct, and Dr. Netherby Winslow co-directs, and then the PEAK lab, which is directed by Dr. Mushton, and Adam is the main lab manager. And so we have done studies like yoga for cancer-related insomnia. We've also looked at the impact of yoga on things like circadian function and looking at RNA sequencing gene changes, which is a really interesting way of trying to understand what's the mechanism behind the intervention. We've also looked at relationships between immune cell function and cognitive decline that sometimes happens in some patients that undergo cancer treatment, and then how exercise can help mitigate those cognitive changes by altering immune cell phenotyping. We've done and have a study that's going on now in cancer-related cachexia, where we're trying to phenotype and understand different clinical and biologic trajectories of how this manifests in patients. And we're very interested in health equity as part of the work that we do. And as one example of that, we're looking at work in disparities in patients receiving immune checkpoint inhibitors and the biologic impact of adverse events in different patient populations. Like sticks. So we have a proven track record of successfully assisting investigators, so both new investigators and more experienced investigators, in securing internal and external funding. We've worked and supported several career development awards, several RO1s, large infrastructure awards like our NCORE research base, as well as training grants like the T32. So high-level overview, before Colleen and Adam really dive into some of the specifics, is that the PEAK side of things supports investigators in conducting multidisciplinary and translational research really to all forms of human movement. And it's really cool to see how this has expanded over the years. They really focus in human physiological assessment as one arm of the PEAK lab. And then the other arm of the PEAK lab focuses on behavioral intervention development and implementation, and also helps train others on how to deliver interventions, and both in person and remotely. And then the CCPL side of things, we've focused a lot. We have a longstanding history in looking at cognition, but also other patient-reported outcomes like fatigue and other outcomes that are relevant to patients as they're undergoing treatment. We have major emphasis in biospecimen processing and kit making. And also we have multiple approaches to assessing biologic outcomes. We have Luminex technology, which we use here frequently. And then we also have a Simoa, which is a unique instrument that I'm not sure of another one that exists in New York State outside of maybe the New York City. So again, we also are working to do remote assessments where we have some innovative things that Dr. Netherby will also be talking about. Again, we focus in multiple aspects of study design, really from like all the way at the beginning where you have an idea. So we help consult with that, and then we help execute it and help with the grant application preparation. And then when you get your summary statement, we'll help look at the reviews and how to improve it. And I already talked a bit about some of the services that are listed here. We have unique expertise, again, combining that physiology and biologic aspect together. And we'll be talking a bit more about some of those things. This is just, again, kind of summarizing some aspects of things I've talked about where we really take somebody from the beginning where they have an idea and try to help them get an end product within the scope of our shared resource. And so, Adam? Thank you.
All right, hello everyone. My name is Adam Sesbikowski. As Dr. Janelson said, I manage the PEAK lab, part of the HBSR portion that I'll be talking to you about. So who we are directed by Dr. Karen Mushton. Assistant Director and HBSR Service Leader is Dr. Poju Lin. As mentioned, I manage the lab, and we have Brittany, Alicia, Marcus, and Cheyenne, all PEAK lab staff assisting us in our day-to-day operation. Assessments that we perform, aerobic capacity, an umbrella term. We do VO2 max testing and 6-minute walk testing. VO2 max testing is done on a metabolic cart shown in the largest picture off to the left. And that is done on metabolic cart, most often done on a treadmill or a bicycle. We also do 6-minute walk testing, which is a submaximal version of a VO2 test, where it's an estimation of their VO2 rather than a direct measurement on a metabolic cart. Pulmonary function testing for investigators that want to know how people's lungs are functioning. Oftentimes, specifically in our work, chemo or radiation can cause damage to the lung tissue. So we want to make sure that, or the PI rather, would want to make sure that their pulmonary function is up to par, or at least want to measure the effect of their treatment on their pulmonary function. I do resting energy expenditure as well, and 12-lead ECG. These pictures here show all the things I discussed. Continued with assessments that we perform, strength and endurance testing done on the Biodex. The Biodex is a isokinetic dynamometer, which basically just means that the machine will move at a programmed speed no matter how hard you're pushing against it, whether it's 4 pounds of force or 4,000 pounds of force. It has been used in hundreds of clinical trials to assess different parameters of strength, torque, work, all measured in foot-pounds or Newton meters. Surface electromyography, that is used to assess the muscle activity measured in millivolts. So from Physiology 101, every time a muscle contracts, it gives off an electrical impulse. What the electromyography does is it measures the amplitude of that impulse in millivolts. We usually put it on large muscle groups like on the quadriceps or the hamstring during a leg exercise, or on the upper body for an upper body strength measurement with the Biodex. They work in sync, the EMG and the Biodex together. BIA, bioelectrical impedance, is a body composition assessment. That tool has over the years become more portable, which is always nice. It assesses different portions of your body composition. So we're all made up of the same stuff, just at different quantities, whether it's bone, muscle, water, fat. The BIA is able to assist investigators looking at all things body composition. Another important tool, and especially in the field of cachexia, like Dr. Janelson's talked about, cachexia is defined as a large amount of weight loss, often seen in some advanced types of cancer. So a body composition assessment, such as the bioelectrical impedance, really aids investigators to see exactly how much and what type of tissues maybe the person is losing. Hopefully not so much. Wearable technology. So this is an area where Peak likes to hang its hat, as it were. Varieties of activity trackers, actigraphs, Garmin, Fitbit, pedometers. People who aren't so familiar with wearable technology, you'll probably see Garmin and Fitbit, and that rings a bell in your head. So some things on here, maybe not so much, are actigraphs. So you can think of an actigraph as a wrist or a waist-worn research grade activity tracker. So it's kind of like an Apple Watch, but just research grade and a lot more precise on how much physical activity the person is doing. Locally and nationwide, our actigraphs are used for daytime wearing for your physical activity level, how sedentary are you, how physically active are you, and overnight for sleep. If an investigator is looking at somebody's sleep quality, the person can wear the wrist worn, more often than not, actigraph overnight, and we're able to do some analyses on the back end to see how well or unwell that person is sleeping. Heart rate variability, that's the bottom right picture. That is a non-invasive way to measure heart rate variability. Heart rate variability is just the variability between your R spikes on your ECG. So R spikes, you can think of it as your heartbeat, and the variability in between those heartbeats is in milliseconds. It's very small, but it is a great tool that we use, again, both locally and nationwide. And it can point to overall health, well-being, CNS processing, things like that. G-Walk motion sensor, that's the bottom left picture. We use that for gait analysis, oftentimes coupled with the 6-minute walk test here locally. We can see how the person is walking, how efficiently they're walking in their gait. When placed at L5S1, like it is in that picture, we're able to see pelvic kinematics. So how the pelvis is moving, how much obliquity, how much tilt, how much rotation, propulsion, acceleration, deceleration, all these different kinematic data can tell us how efficiently somebody is walking. So maybe in a trial where they're assessing their 6-minute walk test at baseline and post-intervention, maybe the participant doesn't significantly increase their distance walked, but maybe they increase their walking efficiency, which might be great for an investigator to point to and say, hey, if our participants are walking more efficiently, maybe they're at a decreased risk for slips, trips, and falls, things like that. The VO2 master is the picture in the upper right. Let me see if I can move this window a little bit. Yeah, upper right, VO2 master. That is a portable cardiopulmonary assessment tool that we are hoping to implement. We'll talk a bit about it more in depth in future slides here. But basically, it does the same things as our big metabolic cart, but in a nice portable, shippable vehicle, if you will. Behavioral intervention delivery. Behavioral interventions with in-person and remote/virtual capability. Since COVID, Peak has really tried to have strength in the field of remote and virtual capability, looking to decentralize intervention as much as we can. Exercise interventions such as XCAP, exercise for cancer patients. Yoga interventions such as the yoga for cancer survivors. education interventions such as the survivorship health education and healthy eating and learning. These are all interventions that we use both locally and nationally in our NCORE trials, and all have the ability to be done remotely or virtually. Hybrid assessments, so assessments that can be done in a combination of in-person and virtual. Cardiopulmonary assessment, like the portable VO2 analyzer that I introduced just a couple slides before. Again, it's a device where we are hoping to be able to ship it out to sites for our nationwide trials and be able to collect remote cardiopulmonary data, whether it's a VO2 assessment like a 6-minute walk test or even a more rigorous VO2 max protocol. Muscular strength and assessments like the 30-second arm curl, the 30-second chair stand, those assessments are used currently in that nationwide cachexia trial that, again, Dr. Janelson's introduced. This picture section on the bottom left here shows a Zoom meeting being held between me and mock participants on this trial, where I would walk them through conducting those two assessments, the 30-second arm curl and the 30-second chair stand, as well as the timed up and go, actually. Heart rate variability that we talked about a few slides ago as well, and physical function assessments, timed up and go, again, the short physical performance battery. Budget estimate, so a PI who is interested in using peak services, the budget estimate that you receive entails all these different things: study startup costs, startup meetings and SOP creation, sample size, how many participants are you accruing, assessments, are you looking at a body composition assessment, are you looking at cardiopulmonary assessment, are you looking at strength, heart rate variability, so on and so forth. And we can always assist and talk you through what might be good for your trial, what might not be good for your trial, taking in time and budget considerations with your funding. Time points, are you looking at a 2 time point, like a base and a post intervention? Is there a midpoint? Is there follow-up 6 months, a year? All those things come into account when creating a budget. Intervention materials, well, we need to order and supply your participants with those wearables, kits for the X-cap or the yoga. Intervention delivery, are you having peak deliver assessments, things like that, training materials and staff training sessions as well. The Peak Lab hourly rate is $188, whereas Wilmot members get a 15% discount. How to access Peak services. Investigators interested in utilizing Peak can email us at our Peak Lab inbox, peaklabexercise@urmc.rochester.edu. We will set up a startup meeting with you and with the Peak Director, Dr. Karen Mustian, and the HBSR Service Leader, Dr. Poju Lin. Follow-up meetings and discussions to finalize study procedures, training materials and training sessions, intervention delivery, data collection, and budget estimates. Your grant will then be billed at the Peak Lab hourly rate based on the services provided by us. And that's all I have. I'll pass it over to Dr. Rizzo. Thank you.
Thank you, Adam. So now we're just going to switch to the other arm of our shared resource, and that's the Cancer Control and Psychoneuroimmunology Lab. And as Dr. Janelson introduced earlier, we're specifically involved in supporting clinical studies and empowering clinical researchers to include biologic correlates in their supportive care trials, who don't necessarily have their own wet lab space to do so themselves. So we are a big collaborative team. We're made up of Dr. Janelson's our director. I myself am the co-director. I have a background in immunology. We have a fantastic longstanding team of technicians who manage everything from coordinator support to biobanking to actually running the immunological assays that we do. And then we also have a fantastic clinical coordinator team who can offer support in how to start up these studies, guidance on how to open studies locally, and things like that. So first, I just wanted to talk about some of the sample repertoire that we have currently in the CCPL. So we've worked with a number of samples over the years, and what's important to note is that this sample repertoire has grown with investigator need. So, when the lab was initially developed, it was primarily just looking at blood samples processed into serum and plasma. But over the years, we've expanded into PBMCs, using whole blood for genetic and genomic applications downstream. We've also developed DNA extraction techniques from samples like bone marrow and saliva. We're collecting urine for studies that are interested in looking at radiation toxicities. We're also collecting stool on local and national trials as well for microbiome analysis and metabolites. I mentioned that we are collecting saliva primarily for ancestry and biologic aid. And then we also do have some collaborators in neuroscience due to the specialized technique or specialized capabilities of some of our biomarker platforms. And we've been able to use this multiplexing approach in cerebrospinal fluid as well. But again, all of these were built out of investigator needs, and we are very familiar with optimizing new standard operating procedures for new sample types. So if there's something that anyone's interested in looking at in their samples or any samples that they have historically stored from previous trials, we have all the resources and expertise to sort of optimize our platforms to be able to offer our services. So we also have some specialized equipment. You can see in this table, the MAPI system is a micro-aliquoting system, so that samples that are collected, it minimizes waste and maximizes freezer storage space. So rather than storing a plasma sample, for instance, in a 2 ml microfuge tube, it can be aliquoted down into 1, 2, and 500 microliter samples and then stored in these customized goblets. And all of these are heat sealed and barcoded so that we can store them for long-term safely in our -80 storage or in our liquid nitrogen tanks. But again, we are wasting less samples and minimizing the number of freeze thaws. We also have 2 Luminex systems, which anybody with a wet lab may be familiar with these platforms. But again, these are available to all of our clinical researchers who are processing samples in our lab or storing samples in our lab. We can do this multiplexing technology, and we have tons of experience doing complex Luminex projects for our users. We also have a QIAcube for DNA extraction, and we've worked really closely with the GRC to optimize protocols for different sample types like bone marrow and saliva, as well as whole blood. SpectraMax, we use spectrophotometers for ELISA and DNA quantification. We also have the Quanterix Simoa, which is the platform that Dr. Janelson's talked about. This is a high sensitivity platform that can detect targets down to the femtomole range. It was really developed with neuroscience researchers in mind and for targets that are higher abundance in the cerebral spinal fluid, but very low abundance in the serum and plasma. So this platform was developed almost as a digital ELISA to be able to detect some of those CSF proteins in the systemic sample types. And then we also have an enormous cache of -80 freezers because we serve as a biobank for the NCORE research base. And then for all of the local investigators who are running their studies through the HBSR, we also have the capability of storing and tracking their samples in our sample management system. So I just wanted to go through a brief pipeline of how a study could start up in our lab. So we do have the expertise in making study-specific blood draw kits. So you can see an example of that right up on the left here, where in our initial startup meetings, we might talk about what the goals are of the PI, what are the primary outcomes that they're interested in, and then to maximize the usefulness of the samples they're taking and also minimize the amount of blood that a participant has to give, we can advise on which types of vacutainers you should draw, the types of processing that might be best to meet your end goals. We can then package these blood draw kits, create labels that are barcoded and can be scanned into our freezer management system, and then track your samples throughout the life of your study. So below that is just a screen grab of our FreezerWorks management system. So again, all of our samples and the aliquots are barcoded, so everyone's scanned in individually. They're assigned a freezer position, a box, a rack, so we know where every aliquot is all the time throughout the length of the study. This can be accessed by our data team for auditing purposes and things like that. And we have in the order of tens of thousands of aliquots of these samples. And then these are just some of the applications that we use most routinely in labs. So typically we are getting blood samples from coordinators and participants. We can either process immune cells from them to do single cell genomics approaches in collaboration with the GRC, or to do flow cytometry in collaboration with the flow cytometry shared resource. Like Guy mentioned, that we have the capability of extracting DNA from these samples, and again, collaborating with the GRC for either genomics or epigenetic arrays. We also very routinely do ELISA, Luminex, and Simoa assays. So sort of like the Peak Lab, when you engage a project with CCPL, we can set up a budget estimate for your project that you can use either for your grant application or just to estimate the overall cost. This typically includes a small amount of study startup, but we are billed using the PPMS kind of a calendar system. So it's basically built on technician time or time and effort spent on the project. Our rate is $174 an hour, but Wilmot members get a 15% discount. And again, I just wanted to offer a few examples of estimates for how much a project would cost. Simply for a Luminex plate, it might be about 10 hours a plate. So you can extrapolate that based on the number of samples and time points and things like that to get a rough estimate of what a project with us might cost. But in the initial meeting stages, you can meet with myself, Dr. Janelsons, and also our entire lab team, and we can set up a project estimate for you. And then I wanted to revisit this cachexia study that we've talked about throughout our presentation. So Dr. Janelsons introduced this, and then Adam talked about this as well. This is a good example of a study that the HBSR was involved in really from the ground up. Dr. Dunn's research idea was predicated upon the idea that cachexia, which is a wasting syndrome in participants with advanced cancer, is usually all lumped into one diagnostic criteria. However, it is likely that there are subtypes of different ways of developing cachexia. And so this longitudinal study was born under the idea that he wanted to better identify subtypes of cachexia and understand them longitudinally over the course of cancer treatment. So in the beginning, Dr. Dunn approached the HBSR, and the Peak Lab got very involved in understanding the body composition and the physical function assessments that they wanted to do in the study. The CCPL also helped identify which blood samples would be most advantageous for them with their long-term goals. The study is actually also collecting tumor types. So we developed procedures to store tumors long-term, and we are collaborating with the Biobank Shared Resource for downstream analysis of those tumor blocks that we've received as part of the study. During the protocol development, we consulted on putting the actual protocol together. We wrote various sections for the protocol. We developed training materials for the external sites. And so Adam and myself and the Peak and CCPL team have trained hundreds of coordinators on this study for how to process blood samples and how to deliver the physical function assessments remotely or in person. So this is one of the hybrid trial designs that Adam had mentioned. So participants here have the opportunity to either do their assessments at home or in clinic with their coordinator. Again, we developed all the training material. We're continually helping with amendments for the study. The Peak Lab is managing all of the actograph data that is being collected as part of the study, and the CCPL is managing the biobanking that we need for this study, and will be performing downstream inflammation analysis and coordinating with several external and internal labs and shared resources to distribute the samples for their downstream analysis. And then I also just wanted to present some of our survey results, which have been really important specifically in our shared resource because we are unique and we can tailor our services more to people. We survey our users every year, and this is ultimately just to say that most of our users are in the cancer prevention and control track at the Cancer Center. And they've been highly satisfied with the work that we've done, and the majority of our users are using both Peak and CCPL together because we really do focus on integrating our services, and that's one of the unique strengths of this shared resource is that we can enable these biological correlates and also integrate with highly sophisticated physical or human performance data to have the end product for these studies. And then we do have some strategic directions. And again, these are really all born from our user needs. It's important to us that we want to collaborate more with the basic research departments and facilitate intra-programmatic collaboration. We also want to further increase our sample repertoire, which we've already started to do this year. I talked about how that's expanded a little bit. And we also want to expand remote and decentralized intervention delivery. Like Adam had talked about, the Peak Lab is always exploring new wearable devices and testing them out. I've even seen Dr. Lin wearing 3 different types of activity trackers at once for like an informal analysis, so that they really do have the most expertise on what would be best for your trial. And so again, the Peak Lab is exploring more technology for virtual intervention delivery, and they are in the process of developing a Peak-specific intervention delivery space. And so With that, I just wanted to share some contact information. We're all very, very accessible. So if anyone needs to engage a new project, you can just reach out to myself, Brian and Lou, if you wanted to start a project that involved some blood processing. PJ or Dr. Lynn and Adam would be involved in any of the Peak Lab studies that you're interested in, or even if you just want to learn more about the things that Peak Lab is doing, I'm sure you could reach out to them. Again, we're pretty easy to get in contact with. And so I think that's about it. So if anyone has any questions, let me know.
And I just wanted to mention also, I forgot to say this at the beginning, but Dr. Lynn is out of the country right now, so unfortunately she wasn't able to be here with us. So yeah, so that's us in a nutshell, big picture. What questions do people have? Any questions? Anybody thinking about doing a project with the shared resource, or just here to learn, or?
Okay, if you came for the lunch too. It's outside. It was delayed.
But yeah, I mean, I know you know someone who's interested in the piece, right?
I guess I just have a general shape of an idea. As my background, as we talked about before, is in head and neck cancer, which of course is intimately related with issues of malnutrition and cachexia, both pre and post treatment. And so I'm kind of interested in developing a project looking at nutritional interventions, but that we can tie back to particular biological markers that go beyond just kind of albumin or, the kind of really historic or broad kind of approaches. And so the idea that we can integrate various assessment techniques is really appealing.
Yeah.
And I think it's just a matter of work on my end to kind of hone down a little bit more into specifics.
We definitely have new nutritional interventions that we worked on
Yeah.
in the past. There was a whole food plant-based diet intervention that we worked on with the T32 fellow in the division. There's a time window eating intervention we've worked on, a vitamin D supplementation intervention that we've worked on. So we have some of that, the nuts and bolts of what you might need for things like that protocol-wise. And we certainly have experience in that area.
Dr. Lynn's also a registered dietitian
Yeah.
as well, so I'm sure she'd have great insight for you.
Yeah, I was gonna say, I would definitely reach out to Dr. Lynn.
Yeah.
And the other person I can think of, just based on the examples you gave, is Dr. Papone.
Mhm.
So he's done a bunch of intervention studies specifically in cancer, patients with cancer. The exact ones that you.
Yeah, certainly.
so-
I mean, we have a new supplement that.
PJ work together closely too,
Yeah,
so.
we have a new supplementation, Jeremy, another T32 fellows supplementation study with a fucoid supplement
Yeah.
for
Gotcha.
fatigue
And then and then, I mean, you said nutrition, but, another one, so, Dr. Gilmore, is Nikisha, she's looking at, green tea extract, EGCG for, cancer-related frailty.
Gotcha.
So we've helped investigators like on the really diet end of things to like supplement end. And I think that that's another novel piece about us is that like our leadership and team is also like experts in the area of these types of assessments and interventions. And so have knowledge of how to also get these things like up and running and also with funding over time, so.
and also with the various patient-reported outcomes that might be important on your study, depending on if you want to look at fatigue, frailty, things like that, physical function.
Gotcha.
We have collaborators and leaders who know which PROs would satisfy those things.
Sounds good.
Sounds good. So set up a meeting.
Yeah, will do.
Sounds good. Anybody else? Questions? All right.
This is part of a monthly recurring series. Next month will be on decreasing barriers to data analytics, highly focusing on our informatics team. A lot of genomics applications, but there will be
Tracy, I'll put that slide back up.
thank you. A lot of listserv emails. I hit up a lot of the clinical departments and then a lot of the PhD programs. But if you don't hear about it anywhere else, it is on the Shared Resources News and Events website. I just have to update the upcoming title, but it's on November 12th. And then the food is set up so that you can grab your plate and then like move down so you can get your sandwich last. But there is plenty if you want to take two. There's 2 trays. Well, maybe start with, I don't know, it's fine.
Thank you for setting all of this up, Tracy.
Thank you,
Yeah.
Tracy.
Sorry the food was late, our delivery driver.
That's okay. No, thank you. No, it was good. Yep.
Thanks for attending.
Thank you.
Thank you again for being here.